Hi, everybody. My name is Hillary Peterson. I'm the president of the Maine Entomological Society. I'm just going to share a short presentation. All right, can everybody see that?
Yes.
Great. Let's see here. So first of all, thank you so much to Bob Nelson, who put this presentation together. So this is just a quick intro to Maine Entomological Society for anyone new here tonight. So what is the Maine Entomological Society? It's a group of about 170 individuals from throughout the state of Maine, including some from away as well, who are interested in the insects and other terrestrial arthropods like spiders, mites, ticks that are found in the state of Maine. We were founded in 1997 at what is now Viles Arboretum in Augusta, and 10 of the original 14 founders are still active. We function as a common clearinghouse for information about Maine's amazing insect fauna. Why do we exist? Most of our members are highly interested amateurs, including many Maine master naturalists. We provide a general forum for interaction and exchange of ideas, as you just heard before I started this. We also do have professionals as members, but most professionals and academics are more focused into a specific insect pest. We are not here to really talk about pests because pests are really maybe 50 to 75%, not percent, 50 to 75 of the species statewide, whereas we have somewhere around 20,000 species. It's probably even way more than that. So this is a forum to celebrate biodiversity, not a pest control forum. We publish a quarterly newsletter with news and views on Maine insects. That's called the Maine Entomologist. And on our website, if you just Google Maine Entomological Society or mainentosociety.org, I think it's mainentomologicalsociety.org, you can find archives back to the beginning of the Maine Entomologist. We also conduct monthly field days from late spring through the early fall and much more. If you are a member and you get the newsletter, the section shows all of that. You can also find our events on the website. This is our website here, again, mainentomologicalsociety.org, and it includes archives of our past webinars. These webinars are scheduled throughout the winter months, so we're like, wow, we're having fun in the summer. We need to have fun in the winter too. We also have a Facebook page. At this point, I think there's over 7,000 members actually, which is awesome. And we also have a modest scholarship fund that can help fund Maine-based insect studies. We've sponsored 13 entomological bioblitzes at Acadia National Park, adding many hundreds of species to the known fauna in the park, including numerous first state records. And members have been key players in 4 major faunal inventories in the state, including the Maine Damselfly and Dragonfly Survey, the Maine Dragonfly Survey, the Maine Bumblebee Atlas, and the Maine Carabid Project. Members have also spent thousands of hours volunteering at the Maine State Museum, working with the state insect collection. We also provided help in a project that resulted in the pink edge sulfur being designated as Maine's official state butterfly. If you would like to donate, especially to our scholarship fund, you can take a picture of this QR code or find the donation form on the website. And up next month on Thursday, April 11th from 7 to 8 p.m. will be Aquatic Insects, Lifestyle, Behavior, and Fly Tying by Eric Fromberg. All right, I'm going to pass it off to you, Roger, to introduce Shyloh once I stop sharing.
All right, and before I forget, Shyloh, I just have to ask you, would you prefer questions saved to the end or taken as they come in?
Let's save questions for the end, just so I can be sure that I get through all the things I want you all to know about tonight.
Sure, okay.
I'll try to move quick enough so that we have enough time to have a good discussion.
Sure. So anyhow, you've just met Shyloh Favreau. It's my pleasure to introduce him. He's the program manager for the Food and Agricultural team at the University of New Hampshire Cooperative Extension. Doing this, he oversees soil testing and insect identification and supports the university's tick education program. Those are all important. In addition, while working full-time, he's currently getting a master's degree at UNH's agroecology lab, specializing in melitology. I didn't know what that was till you told me, but it's the study of wild bees. And he's been particularly interested in how wild bees can be used as possibly used to replace pollinators or add to the pollinators that we use, the honeybees that we have to import in order to pollinate monocultures of food crops. So without further ado, I want to hand it over to Shyloh, and I'm thrilled to hear what you're about to talk about, because it's certainly an area that interests all of us.
Thank you, Roger. All right, let's see. Let me successfully share my screen with everybody. Okay, go. Also, everyone can hear me just fine. My volume is good.
Yes.
Yes.
Okay. Okay. And that's showing up.
Yes. Looks great.
Okay. Again, so thank you, Roger. So for the next 45 minutes or so, I would like to share with you all the Live Free and Pollinate project that I've been working on as a part of my master's thesis. And although this project is based here in New Hampshire, I think that you will find that much of this can apply to Maine and the Northeast overall. And this project, it really has a lot of layers, but the big picture is it aims to examine the wild bee communities and apple orchards across the Granite State. And this project, it's a work in progress. So tonight, I'm really going to focus on giving you some background about apples in New Hampshire and bees. I'm going to walk you through the field season. And though we haven't run any statistical analysis yet, I will be able to share with you some of the bees that have been reoccurring as I've been kind of sorting through and identifying all the bees that we collected. There may be a few quiz questions throughout, so stay alert. So without further ado, I want to jump right in. I'm going to get started with the apple part of this. There are 271 farms growing apples here in New Hampshire, making them the 5th most grown crop here, falling behind hay, corn, mixed vegetables, and Christmas trees. The total acreage currently being used for apple production here is 1,435 acres. And almost all of those acres, 1,239, are bearing acres, which just means that they're currently producing fruit. And according to the New Hampshire Fruit Growers Association, New Hampshire grows over 50 different varieties of apple, which is really great. And recently, I've been reading through the most recent 2022 USDA Census of Agriculture, and that was just released in February. I think it comes out every 6 years or so. So the last data that was published before this was 2017. And in combination with the data from that, I was also using the 2021 National Agricultural Statistics Service. And with all of that information, I was able to do some math. And I found that New Hampshire brings in over $19 million annually in apple production alone. These numbers they're definitely approximate, but I think they do a good job of highlighting the importance of apples here in the Granite State. Okay, so on to the bees. Like all insects, the world of bees is massive, and you all would have to sit and listen to me for hours to even begin to scratch the surface. So instead, I tried to focus on what I think is the most important for you to know about bees as it relates to the scope of this project. I also wanted to be sure that I gear this presentation for a general audience. So if you're new to bees, don't worry. And on the other hand, if you do have a background in bees, I still hope that there are a few things sprinkled in here that you didn't know. And you're probably all familiar with the honeybee, but those are just one of 20,000 known bee species in the world. And 4,000 of those bee species are native to the United States. In fact, there are more bee species in the world than all species of birds and mammals combined. Taxonomically speaking, bees fall under the order Hymenoptera, and that includes many species of bees, wasps, hornets, sawflies, and ants. Under this umbrella, bees belong to the superfamily Apoidea and are classified into 7 different families. And you can see here on the slide that I've outlined the different families here in blue on this phylogenetic tree. So as follows, these 7 families are Meletidae, Apidae, Megachilidae, Andrenidae, Halictidae, Stenotrididae, and Callididae. For time's sake, I'm not going to dive into each individual family, but I will talk more in detail about a few bees in some of these families as it relates to apple pollination towards the end. Essentially, bees are divided into these families based off of shared traits and their evolutionary histories. Does anybody in the audience have any guesses on how many bee species you think have been found here in New Hampshire? Feel free to shout it out.
I think it's about 55.
55. Okay, we have one for 55. Any other guesses?
Actually, 55 is per average location, maybe. It might be more than that in the state.
Okay. All right, so no other guesses.
70.
70. Okay. Owen says 70.
100.
Hillary 100. Okay. All great guesses. Now, I'm going to leave you hanging here for a second. Before I tell you the answer, remember how I told you in the previous slide that the US has about 4,000 different species of bees? Well, the United States actually has the most species present compared to the rest of the world. But depending on the location, there are some areas in the US that have more species richness than others. And when I say richness, I just mean the number of species within a given region. And take a look at this map here and notice the color gradient. It goes from light yellow all the way to black. And the darker the color means the more species richness in that region. And you can see the black down in the southwest, that's where the highest number of bee species are present. And so, for example, in Arizona, there's about 1,300 bee species there. And as you kind of go up to the top where the Northeast is, that color is more orange, meaning less species richness. Bees really love arid temperate regions, but that doesn't mean that we don't have a good amount here. Without further ado, there are 340 bee species that call New Hampshire home. But I think it was that Thomas that you said that about 55 per like area? I think that's
Yep.
like a pretty good, that was a pretty good estimate, I think, at least from what I know.
Mhm.
So not bad. And this is the most comprehensive up-to-date list that I know of currently. I think for Maine, I looked it up today just to be sure. Jennifer Lund, who's your Maine state apiarist, reported in 2021 that Maine had an estimated of 270 bee species. And these lists, they're always changing as new species are discovered in our region, and certainly some of these species are more common than others. So if the honeybee is just one species here in New Hampshire, well, who are all these other bees, you might ask? And well, this list is made up of lots of native and wild bees. Before I go any further, I do want to clarify one thing. You'll notice I use the words wild and native when referring to bees in this presentation, and let me explain why. Not all wild bees are native, and native bees refer to bees that are indigenous to a particular region, and wild simply means that bees that are not domesticated. Most often, native bees are one and the same, but it's good to know the difference. For example, bumblebee, there are many native wild bumblebees, but there are also commercially available bumblebees that have been domesticated and primarily used for greenhouse production. Now, that's a whole different topic of its own too, but the takeaway here is that even though most wild bees are native, there are a few exceptions. Anyways, all these other wild and native bees comes in all sorts of colors, shapes, and sizes. And you can see here on the slide a really extreme example of that. On the left, here is a very small mining bee found in the US, and on the right is a very large carpenter bee, which is like bigger than this quarter. With so much to know about bees, I tried to highlight a few key facts that I think will provide good context for my project. And first, unlike honeybees, wild bees are unmanaged most of the time, and 90% of wild bees live solitary lives. Bumblebees are the only bees that are truly considered social, and they live in colonies, sometimes up to 500 individuals. And some wild bees do live close to each other in larger aggregations, but they're not sharing the same nest. With, again, there's a few exceptions to that too. Speaking of nesting, the second important thing I'd like you to take home is that wild bees do not nest in man-made hives, and instead they construct their own. And 30% of these bees make their nests in cavities of hollow stems. And you can see an example of that on the top. And 70% of these bees make their nests in soil, shown below. Bees also have a variety of floral preferences where they collect their primary diet of pollen and nectar. Some bees are generalists, and that means that they visit a variety of different plants to gather this pollen and nectar. And some are specialists in that they only visit one or a very small number of plants. And the last thing I want to highlight here is their flight range. There are a lot of studies that report the different flight ranges of bees, and some of that has to do with body size and also the landscape. But in general, it's accepted that solitary bees have a short flight range. Again, I'll use bumblebees as an example here, or even a honeybee. Either one of them, their flight range is over a mile, and sometimes even more. But small solitary bees might only be able to travel less than like 500 yards, which is not that far. Again, so there's a lot to know, but I think these points are the most important as we move through this. Now, I've given you a review of apples in New Hampshire and some background on bees. And you might imagine that I want you all to care about this because these two have a really important relationship. Apples are self-incompatible in that an apple cannot pollinate itself. There's a lot of genetics behind this, but essentially pollen must move across different varieties of apples for the trees to produce fruit. And of course, who better equipped to move that pollen around than bees? And as I just highlighted, pollen and nectar are bees' essential food. So it's a win-win, right? This is a really good example of mutualism where both the apple and the bee are benefiting from their interaction with one another. And yeah, it is a win-win, but these days it's a lot more complicated than that. Honeybees have played a big role in apple pollination and in many agricultural systems around the world for a long time. Many farmers manage their own honeybees or they rent them. But honeybees have been declining due to disease and other factors. Since 2006, beekeepers, they lose about 30% of their bees each year. And this has significantly driven up the cost of honeybees for growers. Just curious if there's anyone that has honeybees. I'd be curious if this has been something that you've noticed. And this is where the wild bees come in. The increase in honeybee cost is just one reason attention has turned to the pollination services that wild bees can provide, which is great because research shows that wild bees can be more efficient pollinators of apple and a long list of other agricultural crops as well on a per visit basis than the honeybee, and they do it for free. But due to several factors, including habitat loss and alteration, pesticide use, introduced diseases, and the changing in climate, many wild bees are also facing decline, and at the very least, it impacts their distribution. Now, I don't want you to take away from this presentation that I think honeybees are bad or not needed. Every farm has its own unique situation. I'm just highlighting that there are other bee players on the field that you, and specifically orchardists, can rely on if they know how to support them. And diversity, it's a good thing. the more options an orchard has, the more security they have in getting the pollination services that they need. What I do want to emphasize is that here in New Hampshire, we are known for our small diversified farms and orchards. Many of our orchards here also have other fruit and vegetable crops. And I think this is probably true for Maine as well. Overall, I think it's safe to say that our biggest orchards are considered small in comparison to most states, especially outside of New England. And this is a really unique advantage for us to conserve and improve these wild bee populations. In many cases, simply preserving what we already have is a big part of that conservation piece. Our farms here can serve as great models for conserving wild bee populations and agricultural landscapes and systems. And there's been several studies on wild bee communities and apple orchards in New York that have been very insightful on this topic. And there's also been studies and surveys done in New Hampshire, even some in apples, but these studies have not been conducted statewide, and with small sample size. At least these are the ones that I'm aware of. And that's what brings me to my project. Lots of research and articles are being published all the time, identifying that our insect populations are declining. And as I just highlighted, bees are not exempt from this. And we rely on bees heavily for their pollination services. The USDA states that 1 out of every 3 bites of the food we consume exists thanks to our pollinators, which are primarily bees. I really wanted to build an experiment that would allow me to examine New Hampshire's wild bee communities at the state level, specifically in agricultural settings, and I wanted to choose a crop that was significant and in turn have data that was directly impactful to our farming community. And after a lot of conversations and from my own experiences working in extension, it became clear that apple checked off a lot of those boxes. It relies on bee pollination, it's a significant crop in the state, and I can find an apple orchard in every county here. And the goal was that if I got enough orchard sites and collected enough bees, I would be able to get a good picture of the wild bee communities during apple bloom at each orchard, and then compare those individual communities to one another in hopes to identify some patterns and what might influence or drive those wild bee communities. Is it farm management practices? Is it the surrounding landscape? Is it the size of the orchard? Is it climate, geographic? I hypothesize that more than one of these likely influence the wild bee communities. And aside from being able to contribute to the scientific community with my findings, it really, again, I'll say, like, the extension person in me really cares deeply about delivering this information in an accessible way to orchardists. At the very basic level, I want to be able to provide them with a list of the bees present on their orchard and with guidance on how to support them. Because if you remember from earlier, these bees have a variety of nesting preferences, flight ranges, and identifying these needs is a great first step in their conservation. And hopefully things like orchard management practices will show patterns in the data that I can then use to help orchardists make informed decisions. To me, it's important to take what I learned from bee taxonomy and morphology and apply that directly to an agricultural system that has a positive impact on the farming community. So without further ado, let me walk you through how I conducted this experiment. This field work for my project, it took place last May 2023, where we went out and we collected bees in apple orchards only during the bloom period. And for those of you that don't know, the bloom period for apple happens very early in the spring, starting as early as late April and only lasts a couple of weeks depending on the weather conditions. Honestly, looking back, I have to tell you all, I'm not sure how I managed to pull this off, because on paper you can make a perfect plan, but often things don't always go according to that plan. And preparing for this field season, I thought, great, I'm going to use geography to my advantage, and I'm going to work my way up south to north as the trees start blooming. And in some ways, this did happen, but where my naiveness showed is what I didn't consider that many of those orchards would have trees blooming all at the same time, giving me only a couple of days to visit several of them. And for one person, that's just not possible. However, I was fortunate that my fellow extension field specialist and a group of UNH undergraduate students, and I think one of them is here. I think Santino is here. So on May 5th, 2023, I got the call from the first orchard that full bloom would surely be the next day. And with that, the mad dash for collecting began. In total, we visited 26 orchards in 17 days, representing every New Hampshire county. The numbers you see here on the map represent how many orchards were sampled in that particular county. I mentioned earlier that there have been several apple orchard studies done in New York, and my experimental design was done similarly to one of these studies, or actually two of these studies. Like all research, it's important to make sure that everything is standardized, and therefore bee collection was done exactly the same at every one of these orchard sites. Bees are most active from about 9 a.m. to 4 p.m. And sometimes they're active earlier or later, depending on the weather, but typically that's their time range. Bee activity is best on days that are clear, sunny, and with minimal to no wind. A really good indicator of prime bee collecting time is when you can see your shadow, and that's me, and the shadow makes me look significantly taller. And the best of our ability in such a tight window, we only collected bees during these conditions. At the start of each orchard visit, 3 100-meter transects were identified and measured. One was in the middle of the orchard, one was at the edge of the orchard, and one down a row of Macintosh. These 3 different transects were chosen so that I could not only evaluate which bees were present in the orchard, but if there was a difference in species presence or abundance at different areas of the orchard. Macintosh was the only variety chosen simply because I knew every orchard would have a Macintosh variety, and it provided as a good control. Once these transects were measured, each collector was given a net, a collection jar, and a data sheet. Before collecting, each person filled out this data sheet, which included the collecting event number, location, time, and several questions about the weather during that collecting event. And we used the Network for Environmental and Weather Applications, otherwise known as NUA, weather stations. And for standardization, each person set a timer for 15 minutes and actively used the net to collect bees up and down one of those transects during that 15 minutes. Now, this is where the tricky part comes in, getting all those bees collected over that span of 15 minutes into a jar about the size of a prescription bottle. On a regular bee collecting adventure, I personally am usually focused on collecting maybe 1 or 2 bees at a time, but for this experiment, it was necessary to collect as many bees as possible in the 15-minute event. And especially with a wide range of bee collecting experience, it was the best strategy to get bees into the jar once the 15 minutes was up. Because in most cases, getting the bees transferred to that jar took an equal amount of time. Then all bees were transferred to containers that were labeled with the collecting event, and we sorted them back at the lab later on. You can see here, I don't know how familiar you are with any of the extension field specialists here in New Hampshire, but in the middle here in the red shirt, that's Amy Papineau, and she's our Rockingham County field specialist. And as you can see, she's smiling, all those bees that she just caught. And then over here in the corner, sitting down, that's Heather Bryant, and she's our Grafton County field specialist. And she's very focused on not letting a single bee escape from that net, and she really did a great job. I'm going to try and play the short clip for you all that I recorded after all the bees I collected during an event in my net. Full warning, you might want to turn your volume down if bee buzzing is not a sound that you like. I can definitely tell you that I was stung more times during this field season than all the bee collecting in my whole life. And I've done a lot of bee collecting. So my fingers were very sore. Now, let's see. Yeah, so the bee collecting, it was done at each of the 3 transects 3 times, totaling 9 collecting events for that round. And we had multiple people collecting for most sites every day. So they rotated between the transects. And that was really just meant to help minimize collector bias, because some people are better bee collectors than others. So we just wanted to balance things out. And 2 rounds of these were done at each orchard. So we made sure that one of these rounds was done in the morning and then again in the afternoon, just to ensure that we represented as many bees present throughout the day as possible. And so with 26 orchards, this added up to 468 individual collecting events. One thing I want to mention is that while this active net collecting was happening at each orchard, an additional passive collecting method was employed. During the initial setup, 100 of these yellow pan traps you see here with a flag were evenly placed along the 100-meter transect. And these yellow pan traps, essentially, they're just yellow plastic party bowls filled with 3/4 of the way with water and a small amount of biodegradable dish soap. And the reason for deploying these bowls in addition to net collecting is because these bowls, they're known to collect a lot more of the smaller bees. Where using a net, you might catch more of the larger bees that are easier to see. And so by doing this, I wanted to ensure that we were able to capture as much diversity as possible in such a short window. And there's been a lot of research done in the past with deploying different colored bowls like this, but yellow has been shown to be the most attractive to bees, so we didn't bother using any other colors for this experiment. A funny fact I'll share with all of you is that these yellow bowls, they become harder and harder to purchase because they're so attractive to bees and many other insects. Picnickers and like for parties, they stopped buying them because really it attracted too many unwanted party guests. And you can see on this funny Google image that I found, they're calling it a real picnic panic. So I thought that was funny. Anyways, in a nutshell, this is how we collected bees in just over 2 weeks during apple bloom. And like all research, we counted a few hiccups, but I am happy to report that overall the field season went very successfully. So as you might imagine, we collected a lot of bees, and this slide is a glimpse of what we've been up to. Since May, undergraduate student Lizzie Maurer and I have been busy processing all of the specimens, and we are so close to being done with this process. Each bee gets pinned and labeled with the collecting event number. Then I sort these specimens into morphospecies, and that just means that I'm sorting them by features associated with a particular bee that you can see with the naked eye. For me, this makes the species level identification under the microscope a lot more efficient, so I'm not fishing around for different bees. I can just kind of have a good quick workflow with that. You can see in the middle photo here, the specimens are stored in these wooden boxes, and these will be preserved in UNH collection of insects to refer back to and to use for further research. And on the 2 photos on the side, these are all the bees that we've processed so far and put their collecting event numbers on. And so far, we've processed about 3/4 of the bees that we collected, and so far the count of that is almost 7,000 bee specimens, and that does not include the specimens that were caught in those yellow pan traps. So we caught a lot. The processing, and as you probably can see, pinning and identifying these bees is a long and tedious process, and we are now just starting to get a preliminary picture of who some of those common bees are. Our goal really is to finish up this portion by the end of the semester and spend the summer into the fall running lots of stats and see what kind of patterns come out of the data. And so for the last portion of this presentation, I would like to show you a few bees that I have been most commonly occurring in the species that we have processed so far. But before that, there's one more thing I want to go over. And if you're an entomologist in the group, then this will be a review for you. But if not, this is going to be a lot of information here, but I promise it'll make sense in the end. So just bear with me. Actually, yesterday, I gave an update on this project to the New Hampshire Fruit Growers Association at their annual meeting yesterday, because many of the people in that association are participants in this project. And remember earlier, I said there were 340 bee species in the state of New Hampshire. And so I told them this yesterday. And of course, their response was both exciting, but overwhelming information for them. And I assured them that I would not be sharing 340 different bee profiles with them. And what I explained to them is that it was not necessary for them to remember all of these bees. And that's because, one, all of these bees are not active at the same time. Some bees emerge early in the spring, only for a few weeks, while some bees emerge later in the spring through the summer. And some bees emerge in the spring and remain active all the way through the fall, depending on the latitude and the climate. And two, because apple bloom is so early in the season, this drastically decreases the amount of bee species that will be active during that time. Not to say that most of them don't have other crops, and that they don't need to keep in mind that other bees will be present all throughout the season, but at least they won't have to know, at least they'll know who to focus on during that specific time during apple bloom. The other thing I talked about to them was related to those 7 bee families I mentioned at the beginning. And if you remember, I said that these bees are sorted into these families based off of traits and evolutionary histories. But beyond the families, the sorting gets more specific from there. And you can see from this upside down pyramid of scientific classification, you see where Hymenoptera, the overall order, that includes the wasps, etc. And then below in the green bar here, you can see, sorry. The family, so like Apidae, and that includes bees like honeybees and bumblebees, amongst others. And then underneath that, things get broken up into genera. Honeybees and bumblebees are their own separate genera. And you can see here, Bombus is the genus that bumblebees are in. Now, within that genus, which is Bombus, you get down to the species level, and you can see the example I'm using is Bombus impatiens here. I know that was a lot, and I'm honestly winded myself saying that to all of you. But why I'm telling you all this, and why I told the fruit growers this yesterday, is because when you get down to the species level, in many cases, it's nearly impossible to tell them apart with the naked eye. Using bumblebees as an example again, it's easy to tell a bumblebee apart from other bees, but probably not that easy for most people to tell one bumblebee species apart from another. So I really emphasize to them that if they can get a good grasp of the bees at the generic level, this is in general what they'll need to know to support their bees. And this list, I estimate it brings them down to about needing to know 10 or 15 bee types. Now, with all that in mind, let me show you some of the bees that have been reoccurring species from our field season. In these next 7 slides, the genera name is the first word and the species name is the second in italics. The first bee we found a lot of is Apis mellifera, commonly known as the honeybee. This was not surprising to me, as many orchards have a rent them, or at the very least, someone nearby is keeping them. You all know this bee, so I'll go through this one quickly. The first bee that comes to most people's minds when someone mentions bees, honeybee. They're generalists, they have medium-sized bodies, they live socially in managed hives with several thousand individuals. Their social structure is really complex, including a queen, several drones or males, and worker females. These bees are the most widely used for bee pollination in agricultural systems. I believe there's about actually 8 different species of honeybees, which is interesting because typically you just think, oh, it's just one honeybee. But This is the only one you really need to worry about in the US. The other ones are in Asia and in the Middle East. But one interesting thing about honeybees is the variation in how light to dark the abdomen stripes are. Especially even in the bees that I've collected in all these orchards, some are very dark or some are very light. And that really just comes down to the genetics with who the queen is mating with. Next up, as I've mentioned many times throughout, we have Bombus impatiens, which is a species of bumblebee. And this is just one of 17 bumblebee species recorded in New Hampshire. And in the US, there's just under 50 species total. And surely this is a bee you are probably all familiar with and you've seen. And they're large, hairy, and they're generalists. And Bombus impatiens, it's not the only bumblebee species that I've noticed reoccurring in the bees we collected, but so far it seems to be the most abundant. And they are known to be pretty common around here. Regardless of their species, it's easy to tell the bumblebee from other bees due to their size alone. And bumblebees, they're often the first to emerge in the spring and the last bees out in the fall. Also, bumblebees are always the first up in the morning and the last to go to bed. They generally, they really are the real workhorses of the bee world, in my opinion. And they do this really cool thing called buzz pollination, where they vibrate their bodies to collect pollen and transfer pollen, which is something that honeybees don't do. And I already mentioned this earlier, but bumblebees are social and they live in colonies. And typically they find already created cavities like old mice holes, or if you happen to have an old birdhouse lying on the ground. And if you ever stumbled upon a bumblebee nest, you're lucky because they're really hard to find. And next we have Xylocopa virginica, and the common name is the carpenter bee. There's only 10 species of carpenter bees in the US, but this is really the only one you need to think about, as this is the eastern species. This is the only bee that you might confuse with the bumblebee, but there's a really easy way to tell the difference. And hopefully, see, I can go back for a second. You see this, it's the abdomen. So you can see on this bumblebee, it's very fuzzy. Whereas here in the carpenter bee, it's really hairless and shiny. And these bees are giant generalists. And as their common name suggests, these bees are cavity nesters and they chew large holes in wood for their nests. Sometimes they also nest in dead wood or other plant stems, but typically they're the ones you have to watch out for digging holes in your house. And like bumblebees, these carpenter bees emerge in the spring and they remain active throughout the summer and into the early fall. And moving on to the Serotina species, common name is the small carpenter bee. I've yet to identify what specific species we collected, but I can tell you that we do have a lot of them. And despite the common name, these bees are not closely related to the carpenter bee at all. These bees are generalist cavity nesters, and they prefer to nest in old, like, wood and twigs, as long as they're broken off and kind of decomposed a bit so that they're able to dig through. And a way to tell Serotina is by their small body size. They have little to no hair, and their bodies are darker, and they kind of have this like blue metallic look to them. Actually, because of this metallic coloration, they're easily mistaken for sweat bees, which I'm not showing you tonight, not because we didn't collect any, but because we haven't begun to identify those yet. And they overall had a lower abundance in our samples, at least so far. So we have Calides inequalis. Their common name is the cellophane bee, and there are 13 known species here in New Hampshire. This bee is a generalist, but there are many species of this bee that are specialists. They're solitary, they nest in the ground, and they get their common name for how they construct the walls of their nest, which is something that is really cool. With secretions they make from their own glands and saliva, they line their nest with it. Essentially, it looks like plastic wrap. And these bees can be found throughout the warmer months, but are often one of the first to emerge in the spring. They're considered hairy, and one way to tell a Calides bee is their face structure. It looks a lot like a heart, which I think is really cute. They're a medium-sized bee and similar to the honeybee. I saved my favorite bees for last. I know that I think I sent Roger into a more investigation on these bees when I first mentioned what I was going to be talking about today. And that is Andrena vicina. And their common name is the mining bee. And this species of mining bee is so far the mining bee I've noticed the most as I've begun to identify the bees. However, I can already tell that this species will certainly be one of several species of mining bees I find. And overall, these Andrena bees are by far the most abundant in the bees we collected. I estimate that this makes up about 50% of the bees that we caught, like in total. And Andrena bees, there's more than 15,000 species in the world, and 69 of them are known to be here in New Hampshire. These bees are small to medium, depending on the species, and also depending on the species, can either be generalists or specialists. Their bodies are darker, and in most of the ones we collected, they have dark, shiny abdomens, like you can see the one in this picture. These bees were very noticeable at all of the orchard sites. And at some orchards, their abundance was so high, you would have probably mistaken them for swarms of flies. And many orchardists did think that. Okay, so I said that was the last one. I know we're getting close to time here, but I have one more bee that I want to tell you about. And I swear it's worth it. And that's because these bees, Nomata chrysonii, they are parasitic bees, also known as kleptoparasites or cuckoo bees. And Nomata bees are strongly associated with Andrena mining bees that I just mentioned. And these bees, they don't collect pollen and they don't construct their own nests. And instead, they will sneak into the nest of Andrena bees and lay their eggs. And when the Nomata larvae hatch, they actually attack the Andrena bee larvae and they consume all of their pollen and nectar provisions. I know this sounds really gruesome, but you know, it's all part of nature, right? And in our samples, there seem to be as many Nomata bees as there are Andrena bees. And right now, the majority of them are Nomata chrysonii, which makes sense given that these Andrena mining bees are their primary hosts. And they can be identified by their body shape and color, though in general, Nomata is really difficult to identify. So a lot of the times, even with Nomata chrysonii, it's more of a what you call a species complex, in that probably within Nomata chrysonii, there are different species. They can often be mistaken for wasps. The color is typically red, and you can see here that they have some yellow markings. Sometimes they're all red, and they're fairly hairless. So if you notice that you have Andrena mining bees, it's safe to say that you probably have Nomata present too. And with that, I will wrap things up. But I will say the world of bees is large, and it's a lot to take in and keep track of, especially in this condensed format. But hopefully this presentation has left you with some excitement to support your wild bees. And like I said, this project is still in process, and I will continue to provide updates as I have them, and hopefully publications to share on my findings in the future. I just want to really thank the Maine Entomological Society for inviting me to share my work with you all tonight. It's really great to be around other bug enthusiasts and fellow entomologists. And I'd also want to give a special thanks to all my UNH Extension field specialist colleagues, the undergraduate students, and really a special thanks to all the farmers who welcomed me enthusiastically onto their orchards during a really challenging season. I don't know if you've heard, but there's been a lot of crop loss here in New Hampshire from the weather. I don't know if that also was occurring a lot in Maine. Because without them, this project really would not be possible. So thanks to them, and thanks to all of you for listening.
Shyloh, thanks a million, and an excellent presentation. You're going to have to come back and show us your results.
Yes, in fact, I wonder, I mean, it's really hard to tell by photos on here what these bees really look like. And so, you know, I know that they're New Hampshire bees, but I'm sure you all have your own bees, and I would be more than happy to come up there to do a bee ID workshop sometime in the future.
Oh, that'd be fun.
For that. Yeah, that'll be fun, right?
We'll have to do it before you travel somewhere else for your PhD.
Yes, well, I have family here, so I'll always be around.
All right. So I thought a fun way to actually ask the questions, we have some in the chat, and I'm just going to call on people and you can ask your own question. So Owen Bastille, you're first. If you unmute yourself and show us, and then just ask your question to Shyloh. If you don't want to, I'll do it for you.
Hi,
Yeah, no,
Owen.
I can do this. Hi. Yeah, no, I was probably too early to tell based on what you've said, but can you tell if there are any regions that are particularly diverse or lacking in species richness?
It is hard to tell. There have been some things that I have noticed generally, which honestly, those differences in some of the bees that I didn't mention today, which I didn't highlight mason bees,
Yeah.
and which are common. They're they're earlier, and a lot of them, when I mentioned how some bees are native and wild, but not necessarily always one and the
Yeah.
same. One species of these mason bees, Osmia, they were actually brought here from Asia to help in apple pollination. So I honestly expected to find a lot of these mason bees, and I didn't. In fact, the most of them that I found in the north, which I thought was surprising. I didn't expect that.
Yeah. No, that is interesting. Another thing too is since I also work with Istvan, I was wondering about was are you when collecting the specimens, which net type did you find most effective? Was the sweep nets with the alcohol at the bottom, would that damage the specimen doing something like that rather than having to manhandle them yourself?
Um, when you say alcohol at the bottom, what do you mean
Oh,
exactly?
just in terms of like there's a hole at the end of the net where
Oh,
you
well, I
have the
see.
twist tie and then you release it in there without having to get stung.
Right. No, so we didn't do that. And I'll actually, so when I say the collecting jars that we used, I actually, just due to multiple things. One, it's the best practice for collecting bees. And two, it's
Okay.
humane. We used collecting jars with sodium cyanide at the bottom.
Okay.
And even though we use those yellow hand traps, which are liquid, so wet specimens, um, I ideally in my own collecting, I like to keep the bees dry, because it preserves
Okay.
their hair a lot better. Like they just keeps their,
That makes sense.
as opposed to if you use the bowls, then you have to dry them and it's a whole process. So for that
Yeah.
reason
No.
alone, despite no doubt it was the more difficult choice to,
Yeah. No.
yeah.
Okay,
I
next,
figured you had reasons.
okay.
There
Charlene,
was one
oh,
what was
let's give other folks a chance right
yeah,
now.
no, okay. Yeah,
Charlene,
sorry.
go ahead.
So I was curious about your sweeping method. Uh, were you sweeping the foliage and the flowers of the tree? Were you going after particular specimens, bees that you saw? Were you sweeping the ground? You know, what
Yeah.
kind of technique?
So the technique, so we had all the nets were the same. And what we did was a combination of one thing I made sure that we all did was to be really careful about whacking the blossoms off the tree. Because even though apple growers, they do thinning of those just because, well, there's a lot of that I really can't answer accurately as not an apple grower, but we were really careful about making sure that we weren't whacking the trees. So we only swept up and around the trees, at the base of the trees, and along the ground in that row. And in some cases, if people were feeling bold enough or capable, if they had the collecting jar in their hand and there was a bee right there, and I do this often myself, I just kind of pluck them off and put them in the jar. So it was a variety to make sure that we got as many as possible.
Thank you. Okay, Kathy Claire, you're next.
Yeah, hi. If anybody hasn't said it so far, I like your sweatshirt.
Oh, thank you. Yeah, I got it last year when I was up there helping with the winter workshop.
Yeah, awesome.
I actually get a lot of compliments about this. So you guys did a really good design.
Yeah. So I'm wondering about this parasitism. Do the oh gosh, I can't remember their names now. The adrenids, do they
The nomatobees that parasitize the andrenids.
yeah, do the andrenids, like defend their nests?
You know, that is, it's a really good question. And the thing about this relationship with the nomada and the andrena is there is a lot that we just don't know. We're really in the beginning stages of figuring out that relationship, and if there's like if they specialize in a specific species of andrena. I know that some of the nomada kind of have a few different species that they tend to like. But as far as their interactions and defending, I know that there is, I'm pretty sure from what I know that there's some chemical stuff involved in this so they can sneak in without being seen. But more specific to that, I'm not really sure, at least not yet.
Okay, thanks.
Can I say something about that?
Yes.
Yes, please. If you,
So I have, when I'm taking photos of bees, I very often sit there and watch them for a long time, trying to get a good shot. And I have seen the nomada bees wait like within a foot or so distance to the nesting hole. And then when the andrena bee flies off to go to work, the nomadas sneak in quickly. And they're also very ruggedly built, like most parasitic bees have very strong thoraxes and puckering surfaces that that are very hard, and they can't be stung so easily by the other bee. So they must probably know that the nomadas are not their friends, but they sneak in, and also the nomada bees are pretty resistant to the defenses of the other bee.
yes, thank you for that, Thomas. Yeah, sitting around and being a photographer, you really learn a lot.
That's true. You're an observer.
Yeah.
Yeah.
Are there any other questions? Just unmute yourself if you want to ask a question. So I've got one, Shyloh.
Sure.
Are there any keys that you'd recommend for the amateur to try to identify bees to species or even genus?
Well, I think the genus is manageable. Like I highlighted, bumblebees are big and fluffy. They're pretty obvious. The honeybee, it's not that the honeybee, sometimes, depending on how light or dark they are, if I've moved quickly, I may have put a Calides bee with the honeybee. I've done that before. But, like I think the andrenas are pretty noticeable. It's hard, other than the kind of some of the things that I described there.
Well, what do you use if you have a bee you haven't seen before, what do you use as a key to try to identify it?
Well, at my stage, sometimes I have help by learning a shortcut or two. That's kind of where how I operate, is kind of learning shortcuts from my, I guess, bee mentors, really. Let me think if there's good ways. Coloration is a good way to tell, like you think about a bumblebee. Ways to easily tell bumblebees apart is sometimes where like a band will come down into the abdomen, or some bumblebees they're kind of tricolored, they have like an orange stripe. So there's a lot of coloration things that can be helpful, and even different colorations on the legs or the hair patterns. If you're looking under a microscope, like some andrena, you can tell the difference between lighter or darker hairs. Some of them are lighter or more yellow, and some of them are really they have some really coarse dark hairs on there as well.
Sure. If anybody hasn't seen the book, The Bees in Your Backyard, that's a useful general overview of bees that we're likely to see. around our yards.
It's a really good book. I have it myself.
So
And-
thanks a million. This was a great presentation. There you go.
One. And another really good book, I'll put a plug in this because he's one of my committee members. If you haven't read The Solitary Bees yet, you should. It's a really good book.
Cool. Thanks. I'll
Yeah.
hand this back over to Hillary now, and you want to give any concluding remarks? You're muted anyhow.
Got me. That was fantastic, Shyloh. Thank you so much. You worked really hard on that, and I'm excited to see where this goes.
Thank you. Yeah, I'm sorry. I hope it all made sense. I know you
It
tried
did.
to
It
put
was
all the
great.
information. It's just a lot to take in, but please reach out to me. My email, you can call me. My information is regularly available on the UNH Extension website. If you would like more resources or if you need help identifying a bee, please don't hesitate.
Great. Well, thank you so much, everyone. I think we're going to end things there, and thank you again, Shyloh.